All right. So everyone can see my screen? Thank you for your patience. So my name is Judith Kolkman. This is my cover slide for Z Evolution. I was asked to fill out. I'm kind of a postdoc visiting scientist at Cornell University. A bit of background on myself. I'm from Ontario, Canada. I have a PhD from Michigan State University in bean breeding and genetics, a postdoc with Steve Knapp in sunflower genetics. And I came to Ithaca to work with Tom Britnell at BTI on AC genetics. And I'm working on some of the manuscripts to put out from that before I move into the next position. And then the pandemic hit, and I was just trying to kind of hunker down and get my kids in that and myself through the pandemic. And today's paper is basically my pandemic paper that I'm going to present. My interests are genetics, breeding, stress tolerance. Particularly, I'm interested in GWAS. I really love GWAS and the math game that it is. Interested in agriculture, and I'm interested in world peace. I'm really grateful for all of you in Europe who are coming. I know this is a very stressful time. I have a lot of relatives, extended family in Holland, in the Netherlands, and I know that they're under an extreme amount of stress there. So I appreciate you coming to my talk today. One of my recent papers is looking at a set of nested near-isogenic lines and running GWAS on these lines to show that you can actually use GWAS in a set of nested nils. And I showed that Ligulus 1 is in fact a resistance gene for northern leaf blight. And that was published in G3. He said on the bottom not to hesitate if I'm looking for a job, and yes, I am. I'm writing my manuscripts and I'm looking for the next position. So keep that in mind. If anyone hears something that just send it my way. I'd love to hear about it. So my talk today is a talk that's soon to come to bioRxiv near you. It's called Uncovering the Roles of Lignin-Related Genes in Disease Resistance in Maize Using Mutant and Associated Analysis. This image here is an image of brown midrib corn of no disease and inoculated. And you can kind of very much see what that is. Now, just to acknowledge everyone, I'm in Rebecca Nelson's lab. This paper includes work from Danilo and Ace that helped me score anthracnose stalk rot and anthracnose leaf blight. Peter Bradbury assisted me with wrangling 26 million SNPs. Paul Stokowski is the farm manager who kind of set me off on this project and kind of put me on a tangent for this project, and Natalia De Leon, who supplied some brown midrib mutants that were adaptable to our region. So we are in upstate New York, okay, and this is our field. This upper image here is our field site, and most of the experiments I'm talking about today happened in this circle right here in the fields in this area. If I scan out to the greater upstate New York region, you will notice that there's Ashland Dairy, Sunnyside Dairy, Trinity Farm. This is a large dairy area. New York is a large dairy state, and there are large dairies in this area, 2,000 to 6,000 head of cows. And with that comes a lot of silage corn. And Paul Stokowski once told me, I used to just go to the field and go straight to daycare to pick up my kids when they were little, because no daycare waits for no mom. And when you have to get there when you have to get there. And he said, Judy, you really should start taking the side roads and look at some of this brown midrib corn. And this is an example of brown midrib corn from the Sunnyside, near the Sunnyside Dairy Farm on this field. It's a beautiful field. It's a beautiful region. And you can see all this necrosis on the bottom of this field. This is all northern leaf blight. So this is kind of how this project started, because I didn't know anything about brown midrib mutants before, but they're identified by reddish brownish pigmented midrib and stalk pith. In fact, the whole leaf actually has this pigment, but you only really see it in the veins. It's visible around the 5th or 6th leaf stage of the plants. It's the characteristic is it has reduced lignin content and altered lignin composition, and that makes it popular for dairies for increasing digestibility for cows. So the increased digestibility leads to increased milk production, decreased waste, increased feed conversion. And so that's been very popular. And these brown midrib mutants have been around for a while. I think the first brown midrib mutants were discovered almost 100 years ago. And in the 70s and 80s, they were trying to make them into hybrids. but they've really caught traction in the last 10 years or so, where they've increased the ergonomics to make them more viable. Now, they're characterized by this lignin component, that there's reduced lignin. And here's an example of secondary cell wall, just for review. This is from Sam Hazen's lab at UMass, where you have the cellulose and this lignin compounds, this lignin that's interwoven in between here to provide mechanical strength. If you zoom in on the lignin, lignin is composed of three monolignals that form this polymer. The S and G, S is the purple, G is the maroon, and the H is the red. And most lignins are comprised of the S and G lignin monolignals, with a small amount of the H monolignals as a part of that. And they're derived in the monolignal pathway that feeds off of the core-phenylpropanoid pathway. And I like having this separate because off of the core-phenylpropanoid pathway comes a flavonoid pathway as well. This is from a paper from Gomez-Cano et al. They're Grotwald and John Gray's lab, Michigan State and Cincinnati. And this is a remarkable manuscript that talks about the lignin pathway and flavonoid pathways and implications. And any of you who are interested in learning more about this, this is a really wonderful manuscript. But basically, here's the phenylpropanoid pathway and the various routes to get to S, G, and H monolignals. Now, the first four mutants are the ones that I'm going to talk about early in the first part of the talk. Brown midrib 1, 2, 3, and 4 have been defined, and I'm not going to slander through the names of each of these because this is taped and will be recorded for infinity. But you can see that brown midrib 1 and 3 are further down in the monolignal pathway. Brown midrib 2 and 4 feed into the lignin pathway, so they're before the monolignal pathway. And they have reduced lignin 10 to 20% for 1. Brown midrib 2 is 15 to 25%. Brown midrib 3, 25 to 40, and brown midrib 4 is 14% or 15%. And this reduced lignin has an effect for many reasons, and it's fairly obvious why lignin is important for architecture, for strength, for turgor pressure. You know, it's important for xylem and mechanical strengths. It has a physical barrier for pests, but also is involved in plant growth and development and plant defense responses. So when we first saw these fields, we said, well, it really can't be that simple that you know, you knock out one of these genes and it's more susceptible. And it really turned out to be quite simple like that. Here's the same field about 1 or 2 weeks apart. And during the season, the northern leaf blade just torched up this plant. It was amazing. And there's a wind shear event a week or two later, and you know, there's massive stalk lodging. And brown midrib corn with the reduced lignin is already susceptible to stalk lodging, and you put northern leaf blight on top of it and it makes it even worse. And what this led to was an earlier harvest than normal because of all the senescence. And all the brown necrosis in this field is actually northern leaf blight. Now, I like to brag that northern corn leaf blight is one of the most devastating foliar diseases, the most common foliar diseases. And this is from earlier on in our epidemic. And I think there's about 3 or 4 years where we really had an epidemic in New York and in the Midwest for northern leaf blight. It's one of the most devastating or one of the most common diseases, prevalent diseases. In these more recent years, it's kind of tamed down a little bit. It's still present. The gray leaf spot is certainly taken a leading role. And a lot of that is due to very susceptible germplasm that's been growing. And tar spot. Tar spot is one that should be keeping people up at night. It's been moving across the Midwest, and this past summer it moved into Western New York. So that's one to keep an eye on. That's going to be a problem. Now let's get back to northern leaf blight. Northern leaf blight is a vascular pathogen, and it's a very nice pathogen to look at lignin in because it does target the xylem. It starts with infection. Once it's infected, you have these infection flecks, wilted lesions. They enlarge and coalesce to become these big lesions. And you can see there's a spore producing episorium and infection in the leaf. And here's a beautiful 3D image from Randy Weiser and the NSF plant disease group, where you have a spore that landed on leaf tissue. And the really interesting thing about northern leaf blight is it targets the xylem. And in other studies with the southern leaf blight, and Peter can attest to this too, that the southern leaf blight kind of stays around in the mesophyll a lot longer, but northern leaf blight really does target the xylem, and the xylem's where the lignin is. So that's an interesting point. And here's an example where you have a wilted lesion. It becomes larger, it coalesces, and sometimes you have large epidemics. This came after a hurricane came through and dumped 5 inches of rain, and along with that hurricane came a lot of spores. So they're large cigar-shaped lesions that coalesce. It's a worldwide distribution. It's a worldwide problem, as we heard about a month ago from one of the other talks on C. dyspharia. I like to say that I can go into any field and find northern leaf blight lesions. That's happened almost every year except one where it was extremely dry. And I still found it in my mom's sweet corn field. But you basically can find it everywhere. So there is resistance to northern leaf blight. This is just an example to show the joint linkage mapping in the northern leaf blight in the NAM population from Jesse Poland's work, and where the 3 bars are disease leaf area 1, 2, 3, and an index, which is basically an average of all 3 diseases. So you can see with these dark brown, these dark blue intervals, there are quite a few. I think there are about 23 different QTLs across the genome. In this region, on chromosome 8, we have the HT2, HTN, HT3 gene complex, although we have no evidence for a major resistance gene in this group. It still is interesting that there's something there. I'm going to throw around a few terms throughout this. One is disease leaf area. Just to warn you guys, because I'm going to go when I talk about this, I score in the field 3 times for these experiments. I scored 3 times in the fall. Disease leaf area 1, 2, and 3, about 7 to 10 days apart. And from those, and where I look at the row and I give the entire row a disease leaf area measurement, okay? And then from that, I create an area under disease progress curve, and I divide that by the number of days to make it even. So I have an AUDBC per day kind of measurement. Now, the goal of this, the objectives of this paper and this project were threefold. One is is there loss of function of disease in the brown midrib? Is there susceptibility to disease in the brown midrib mutants? Is there evidence of pleiotropy between lignin and disease resistance? And is there natural variation for lignin-related resistance genes for northern leaf blight via GWAS? And so so I went from the mutants to pleiotropy to doing a case study with northern leaf blight resistance. So first, let's look at the brown midrib mutants. I was able to obtain brown midrib 1 through 4 in a W64A genetic background from Natalia De Leon. And she came here for a talk, and I knew she worked with them, and so she was. It was great that she could send them to us. W64A grew really, it was very well adapted to the New York growing region. And as you can see, the one on the left in this image is wild type, W64A, and this is brown midrib 3. So you can be as objective, try not to be as subjective as you want, but that's a pretty obvious stark difference. And that's from my first year of trials. So what I did was I had replicated trials, 3 reps per trial across multiple years, and I screened these lines for foliar diseases, northern leaf blight, gray leaf spot, anthracnose leaf blight, and Stewart's wilt, which is a foliar bacterial disease. And then I looked at an ear rot and a stalk rot, anthracnose stalk rot and gibberella ear rot. Gibberella ear rot is the more prevalent ear rot up in our region. And here you can see that they all do look kind of the same, some of these. Northern leaf blight, anthracnose, Stewart wilt, and gray leaf spot. These 2 are very difficult to tell, northern leaf blight and Stewart's wilt. And so I really, but they're very different. One's a bacterial pathogen, one's a fungal pathogen. The Stewart's wilt requires a corn flea beetle as a vector to actually get into the plant. And when I scored my lines, I only actually scored Stewart's wilt for the primary leaf area, that initial infection on the leaf, because anytime after, at a certain point, we have so much northern leaf blight in our field that it's just very difficult to score. Gray leaf spot, we grow in the southern tier of New York in a river valley where there's natural inoculum, and it's defined by these really rectangular lesions, which are really remarkable when you see them, how well defined they are. Northern leaf blight targets the xylem. Gray leaf spot tends to be defined by the xylem to make these rectangular lesions. So to look at the results of these brown midrib mutants, I've got W64A in each of these graphs. There's the wild type plus brown midrib 1, 2, 3, and 4. And this is a Dunnett's comparison test between the inbred and each mutant. And it's pretty obvious to see that there's, on the large scale, they're very susceptible to northern leaf blight, gray leaf spot, anthracnose leaf blight, and even Stewart's wilt with the prime. These are all average DLAs for northern leaf blight, gray leaf spot, and anthracnose. Stewart's wilt is just a primary DLA, not the average like the others. So you can see that there's a trend towards being more susceptible. Interestingly, brown midrib 3, which has the least amount of lignin, tends to always be the front runner for susceptibility. And here's an image of brown midrib 1 through 4 for gray leaf spot that I just plucked out of the field. And you can easily see that there's more disease. And so that's for the foliar pathogens. Evidence of susceptibility for stalk and ear rot are not quite as clear cut. The anthracnose stalk rot looks like there's susceptibility at the 0.05 and 0.10% level. And honestly, that's a really good, I feel really good about that because it's a very challenging pathogen to score in the field. You have to take a machete and cut through, and given, I think given another year or two, this probably would have been more susceptible. But so the trend is that these are more susceptible to ear rot, this stalk rot as well. Gibberella ear rot is a fascinating case study because brown midrib 1 and 3, which are what is available in hybrids for commercial use, they are more susceptible to gibberella ear rot and not brown midrib 2 or 4. And that's a fascinating case because brown midrib 2 and 4 have different SG ratios than brown midrib 1 and 3. And if you'll remember, 1 and 3 are further down in the monolignal pathway and 2 and 4 are further up that feed into the monolignal pathway. There are differences in expression of the genes in the monolignal pathway between these mutants, even though they all have reduced lignin at the end of the day. And there's differences in cell wall composition between these mutants. Also, in a study with common smut, brown midrib 2 was found not to be more susceptible, but brown midrib 1, 3, and 4 is. So it is possible that there is some difference. The important thing to notice is that brown midrib 2 and 4 are not actually more resistant, they're just not more susceptible. But this can tell you something about maybe how gibberella ear rot works and what targets you can use for breeding for ear rot. Also, it also tells you that brown midrib 1 and 3 might make for very bad field corn, grain corn to put in your silo. So to spray or not to spray, brown midrib maize is a very susceptible market class, hands down. It has a long way to go in resistance breeding to make up for it. You know, spraying for foliar or fungal diseases can be a very sustainable management tool. Resistance breeding is crucial, crop rotation is imperative. And we want to talk about disease risk management. You saw in an earlier slide that gray leaf spot has really taken off. There are ecological impacts of growing susceptible BMR corn over large acreages because they increase the inoculum load of something that might not be a normal environment for that pathogen. Gray leaf spot likes river valleys, but if the susceptible germplasm is there, it will take advantage of that. The susceptibility to diseases, well, if they may be foliar, they can also cause susceptibility to stalk rot, and those stalks are really what's important for the silage corn. They're what make up the biomass of silage corn. And again, brown midrib corn would make for very lousy silage corn. So the second part, evidence of pleiotropy. And this is, can we find colocalization or correlation between a lignin trait, in this case stalk strength, which is my best vehicle for that, and disease or disease-related traits? So I made a composite graph of the QTLs in the NAM population for Rhine penetrometer readings, stalk strength, versus NLB, GLS, southern leaf blight, lesion mimic, HR, and flecking, or physiological flecking. And these are out of Peter Ballincurdy's group, part of our lab, Jackie Benson in our lab, and a lot of work out of North Carolina, and this is from Jason Pfeiffer and Ed's lab. So in the brown, you've got where the QTLs are for stalk strength. The dark blue is northern leaf blight, the light blue is gray leaf spot, southern leaf blight, and the greens are flecking and lesion mimic. And in the pink along the genome is where the locations are for the brown midrib genes. Okay, so what does this mean? When you look at this, there is really no global pattern. Okay, so it's not like there's something under everything. Not every brown midrib gene has a QTL for stalk strength and northern leaf blight, but there are a few possible co-localizations. This northern leaf blight and brown midrib 2 is awfully close, but it's at the end of the chromosome and it's not overlapping. This stalk strength and this brown midrib is very close, but again, it's not overlapping. There are two, brown midrib 1 and 5 are located here and near an NLB QTL. There's a stalk strength, so maybe that's something. So, you know, there's no global pattern, but it's possible. Then I went further into that and I said, well, what are the correlations within the RILs of each of these traits? So this is a correlation between stalk strength and disease or disease traits, flecking, lesion mimic, HR, northern leaf blight, gray leaf spot, and southern leaf blight in each of the families to stalk strength. And I've highlighted in red the ones that are significant. And what you see in that is, again, there's no global pattern, but there are a few possible correlations. So the answer is there pleiotropic effect? Definitely maybe. And that's my conclusive statement, is that there's definitely maybe an effect. That brings me to the third part of the talk. Can we identify natural variation for resistance to northern leaf blight via GWAS? Are the brown midrib genes contributing to natural variation for northern leaf blight, or are there other lignin-related genes that I can identify? Again, here's going back to the NAM QTL. We know there's plenty of variation for quantitative resistance. And so I looked at the genome-wide, the diversity panel, the Goodman diversity panel, and I screened these over 3 years at the Musgrave Research Farm. GWAS is a numbers game, and I love GWAS, and it has not always loved me back. But I've come to realize it is strictly a numbers game, so I impose some fairly strict metrics to my GWAS analysis. The first is I screened these lines, the Goodman diversity panel, across 3 years in the same environment, 2 reps per year, with the same race 1 of C. diphtheria turcica, which is our local isolate. I phenotyped using 3 DLA measurements and created an AUDPC per day metric. The phenotyping is done by the same person. So I'm the one who phenotyped all of this 3 years, 3 times per year. So I had the same visual, because it is a visual score, which is a percentage, each DLA is a percentage, it's important to have that same bias. People can have different biases, but as long as that same person is measuring at the same time, that helps. Transformed the data for normality and included DTA, data-synthesis, as a covariate for the BLUPs. The GWAS design, I looked at my data, pulled out 273 lines that included at least 2 to 3 years of data. If it only had 1 year of data, I tossed it because I wanted to have it as strictly as tight as I could possibly have it. And I think it's a very important thing to have. I used 23 million SNPs from HapMap version 3.2.1, my allele frequency of 0.05, minimal count of 20, and I use mixed linear models in Tassel. And you can see that I've got a large distribution. I have everything from fairly resistant to fairly susceptible lines. And this is what my Manhattan plot looks like. And I was really quite pleased with this result. And here's a Manhattan plot from chromosome 1 to 10, minus log 10 p-value on the y-axis. And I made an empirical threshold of 5, minus log 10 p-value of 5. Obviously, with 26 million SNPs, you're not going to have your von Fraude correction is going to be too conservative. I think I needed to put the FDR rate down to 70% if it was going to include everything, but not all. I basically outlined and suggested I'm just going to look at anything that's around log p-value of 5 or greater and see where that brings us. And if you look at the QQ plot, I've got really nice control for my p-values, observed versus expected. So I'm very pleased with that. First thing I want to do is look at the brown midrib genes. And here we have graphs of 250 kb up and downstream of each of these genes. These horizontal, or these vertical bars represent the gene space for each of these genes, brown midrib 1, 2, 3, 4, and 5. I ran a von Fraude threshold and FDR in these smaller regions. So well, for the greater Manhattan plot, I just created an empirical threshold. When I went down to 500 kb around these genes, I used von Fraude correction factor and FDR rate. And here you can see, using a more localized threshold for significance, there's no significance in these brown midrib 5 regions, okay? And in these 5 brown midrib mutants. And that means a few things. It could be that there's no variation for resistance to northern leaf blight in the brown midrib mutants. It could be that they're highly conserved. Anything that has that much, that reduces lignin that much, has to be susceptible to adaptation, and so that wouldn't be normally selected for. And a lot of these brown midrib mutants, they're found independently. They spring up in breeding programs, or they're tagged through mutagenesis. So they're not really nicely adapted plants that you'd select for normally, that would survive well, because they have such decreased lignin. So perhaps there's no variation. Perhaps any variation is buried in the minor allele frequency, which is always possible. And it's also possible that any variation is too small to detect. And in that case, my Ligulus 1 is a gene that I identified in the nested nils. And just as a corollary here, and I looked at it in this manuscript for this analysis, it did not hold up. And there's a reason for that, because I think the variation was too small to detect in this panel. So let's have a look at these. What are the rest of these candidate genes? You see this large SNP right up here, the highest SNP in the association. I want you to forget about that SNP. That SNP is a pseudo-SNP. It's heterozygous, and when you look closely in the HapMap file, there's a large block of heterozygous SNP that all segregate together, so it's obviously a duplication. So that SNP is out. What are the next SNPs? And I went through carefully, and I looked at all these SNPs, and I was very excited, and it was like Christmas to find out what you got. And any gene that doesn't have brackets on the around it, the SNPs landed within the gene or very near the gene. By very near, I mean within 1,000 base pairs of the gene. So you can see that there are some fairly nice some hits in here. If it doesn't have a SNP, a gene associated with it, I looked at it and I couldn't easily determine what it could be. If the gene name is in brackets, that is my estimated guess of what the gene is. It's probably in a gene space that's large, doesn't have a lot of genes in it, and that might be the most logical gene that I could assume would be related. So in a big table format, that's what this looks like. And what I want you to see in this is that, this is a table with all the SNP hits and the gene annotations and where they're located and the gene names. Some of them have a lot of clusters, SNP clusters. Some of them have 15, 17 SNPs above a minus log 10 p-value of 5. Some of them only have 1. Okay. And I think that's the beauty of having 26 million SNPs, is that you can capture that 1. The variance explained anywhere from like 8.5 to 15.5. In this case, we have 2 cases where where it hovers around 20%. In both of these cases, there's a lot of insertion deletions in those regions, so I think that's that's an inflated estimate. And some of these are in NAM-QTLs and some of them are not. So I want to focus on the lignin. So I looked at this and said, let's just pull out the lignin-related candidate genes in this diversity panel. And so I pulled out some of the lignin, and I want to go through a case of 6 of these. I want to zoom in on 6 of these genes. And when I do that, I'm going to do 2 things. I'm going to zone in on the GWAS and the LD, but I'm also going to zone out and look at the broader LD structure around those genes to see if those genes are really real or if they're linked to something else. So I established LD across with 1 megabase pair. 1 megabase is up and downstream of the most significant SNP. So I took that most significant SNP and I said, what is the LD of all the SNPs upstream and downstream? So I got this 2 megabase region where you're looking at LD in relation to that most significant SNP association. And then I looked within 500 base pairs. This shaded area here is a 500 base pair region, 250 kb up and downstream of that most significant SNP. And then I compared it with the LD. So these next graphs, and this is an example for the beta glucosidase gene cluster. And these next graphs, this is going to be in the insert at the top to give you an idea of the general area of LD while we focus in on the 500 kb. And here is the strongest SNP cluster that involves a beta glucosidase gene cluster. So here's that LD estimate in that region, that 2 megabase region. And here's a Manhattan plot. The black dots are the GWAS hits. Okay, so that's your normal Manhattan plot. This red dot is the strongest SNP association identified in this region. Okay, Anything that's gray is the LD of the SNPs in association, that in comparison to that SNP in red. So we're looking at the LD and how it decays around that region in comparison to that SNP. These bars here represent 3 genes. This is the gene space of 3 beta-glucosidase genes. And so what you can see here is that these 2 beta-glucosidase genes here have a lot of SNP hits. They're significant SNPs. There's lots of LD within them. Is it this SNP? Is it that SNP? This gene or that gene? I'm not sure. And I wouldn't be able, you can't parse it out from this. But they are, they're beta-glucosidase genes labeled 24 and labeled 25. They're in the coumarin biosynthesis pathway, which is downstream of the phenylpropanoid pathway. They're involved in storage and transport of monolignals, and these 2 genes have homology with an Arabidopsis beta-glucosidase genes that interacts with monolignals. So this actually shows that there is some storage and transport issue with monolignals, and this is one of the strongest gene clusters that we have, providing about 15% of the variation. I want to look at the next one here. This is HCT11. HCT11 is just a beautiful candidate gene. Here's the 2 megabase LD region around the most significant SNP, and you can see that there's really no LD in that area. If I look closer in this 500 kb region, you can see the GWAS hit, and you can see this really nice tower of SNPs right within the HCT11 gene. And that's just tremendously exciting to me. It's also interesting that there's this gap in sequence, which might indicate this presence-absence variation, which could be relevant for the disease resistance. HCT11 as a gene is, so this is really promising. Here's the most significant SNP. And here's a bond-forming correction factor in threshold in black. Gray is a FDR rate. So you can see regionally it's significant. And so I want to look closer at HCT11, because HCT11 is in the monolignal phenylpropanoid pathway. And it is one gene out of a large gene family. It has 42 different genes. And so it really shows that there's a lot of subfunctionalization. There's a huge gene family. And HCT11 is here, down here in this HCT clad 4. And you can see that there is expression. This is from Gomez-Kino et al. There's, contributes about 8% of the HCT clad 4 gene expression. So what does HCT11 do? HCT11 is one of 5 genes that regulate the substrates between the core phenylpropanoid pathway and the monolignal pathway. Okay, so that's really fascinating to me, that this is a gene that's really actually involved within this pathway itself, regulating the substrates that go into the monolignal pathway. And so I looked at those other 5 genes and I said, what are these 5 genes doing? And here's our HCT11 with an obvious peak. And it is the only one of these 5 genes that looks to have an association with disease resistance. It's also a good candidate for multiple biotic and abiotic stress. When I put it back on this pleiotropy co-localization QTL map that I made, is located right here, right where there's an NLB QTL, Rayleigh spot QTL, and a lesion HR kind of QTL from Peter Ballankurdy's group, which is really exciting. And that's also exciting because Peter Ballankurdy, as I'm sure he could tell you, identified HCT1 and HCT2 as genes involved in the autoimmune response in maize. So that's really fascinating because HCT1 and 2 are in this other clade, they're up in clade 2, and mine is in clade 4. So that's fascinating. This is one of my big wins of this entire manuscript, to show that. Two other related traits, or a few other genes. Some of you may have seen, I have NOX1 and NOX2 listed here. And if I look at the NOX family of genes, NOX1 and 2 are the only genes in the family that look like they have any contribution towards NLB resistance. If I look closer in the maps, you can see these are not quite as perfect as HCT11 or the beta-glucosidase genes. You can see there's a huge tower of SNPs near NOX1. These are all in LD with each other. And this is the area of NOX1. It's about 15 kb upstream of this peak. But there is a significant GWAS hit locally. and LD. So this is my candidate gene for this region. There are two other genes closer to NOX1 than this peak. There are no genes in the genome in this peak, and the two that are in between here are uncharacterized proteins. So this remains my candidate gene of choice. The most compelling one, NOX2, there's actually a SNP hit, this red dot right within the NOX2 gene. And as you can see by the greater picture, when you scan out, there's a lot more LD. It's quite messy for LD, especially upstream of the gene. But what remains to be is that this gene is the strongest SNP association. So even though there's lots of LD, this does remain to be the strongest SNP association. And we know that NOX1 and NOX2 are involved in development. They have implications for lignin. Yeah, and so it remains, this remains more of a developmental lignin-related kind of candidate gene in my mind. The other lignin-related candidate genes, ZIM36 is an interesting case where I only have a single SNP. Here in red, the LD is a little messy outside the 500 megabase, 500 kb region, but it remains to be a single SNP. There's no tower, but ZIM36 is a really interesting gene because it targets brown midrib 3, and it specifically targets brown midrib 3. So it's fascinating because it actually might show that there's a role for regulation of brown midrib 3 in defense response. Another, while this is a single SNP, looking like it's all alone, there's another group of SNPs around on chromosome 8, around 80, 81, 82 megabases, base pair megabases. And you see there are a number of SNPs in this region. There's a huge LD block, and I think this is largely driven by insertion deletions in this region. If you scan closer, there's a lot of indels. In my region here, I only really have 4 genes, and LBD35 is in this really slim column here, and here's the most significant gene. I think LBD is a fascinating target because based on the Gomez-Kano et al. Paper, it's a transcription factor that targets the highest number of monolignal phenylpropanoid genes of all the 40 transcription factors that they tested. This LBD35 targets the most lignin-related genes in that pathway. On top of that, even though it's the most compelling gene, it needs to be proven. I found this gene in an NLB cluster. This has also been identified in a diversity panel for in the drought-tolerant maize for Africa panel, which was scored for tar spot. They also found a similar large peak cluster of SNPs, and this gene right here is the LBD35 gene that they identified as a candidate gene. So this is fascinating to me because this is a transcription factor. It may not be the real gene, it might not be the real deal, but it is a very compelling candidate gene. Targets the phenylpropanoid monolignal genes in a region that has low gene content, lots of insertion deletion. So there might be presence-absence variation in there and other genes that we're not aware of. But this region was identified in 2 different diversity panels. And I think that's exciting. It's also a gene that would make sense to have some multiple disease resistance to tar spot. So while I cannot screen for tar spot in New York because it's not here, it would be fascinating and brilliant if by screening for northern leaf blight, I can identify spot resistance genes. So, from those genes, there are a lot of other genes here that I haven't mentioned, and they could be targets for breeding for silage corn as well as for grain corn. So there's a lot of options, a lot of things. Someone said, Judy, you can spend the rest of your life the rest of your career looking at each and every one of those genes, and I think that might be true. So what are my takeaways? My takeaways are, are there multiple disease resistance genes, or is this a disease resistance pathway? And certainly the phenylpropanoid monolignal pathway have a lot of genes, big gene families, and they're sure to have subfunctionalization for disease resistance. And a case in point is this HCT11 is a really good example, because not only was it identified for northern leaf blight and gray leaf spot, but it's also a target EVB protection that Rory Sawyer's group has identified. So this has a potential for abiotic and biotic stress tolerance. Implications: brown midrib genes confirm multiple disease susceptibility, not necessarily resistance, but susceptibility. GWAS can resolve lignin-related candidate genes across a broad swath of functions. So we have some that are developed in development, some that target the brown midrib loci or genes within that family, some that target the monolignals, and some that target the flux between the phenylpropanoid pathway and the monolignal pathway. Yeah, so we have candidate genes for lignin-related resistance genes that have strong implications for other diseases. The exciting thing is to see that HCT11 has multiple disease resistance. The LBD35 has a lot to be proven yet, but it's a good potential for a gene for resistance to both northern leaf blight and tar spot and others. And I think that's the end of my talk. Thank you for your patience.
Okay, thank you for these interesting insights into resistance. We have some questions, discussion already ongoing on the chat. Maybe Jeff and or Arcadio, you want to go ahead and...
Sure. I was just asking about that SNP on chromosome 4 you told us to ignore, that if it's tagging duplication, but that is associated with the phenotype, while it may not be a correct SNP, if it's tagging some structural variant or duplication that is associated with the phenotype, isn't that of interest?
It is of interest. It's yes, you're right. Actually, you're completely right. I probably ignored it just for ease of low-hanging fruit. But it probably should be one that I look into in more depth to see why it's like that. The problem is the LD is so strong across that region that it's either all or nothing. So yeah, that's a very valid point that I should look into more. Thank you.
Yes, and Matt had another question.
Yeah, definitely some really compelling candidate genes within the GWAS hit. So I was just wondering if you looked at whether or not there was overall enrichment of lignin-related genes and the associations with the foliar diseases.
That's a great question. This is kind of there's only so much that you can put in one paper, and there's a lot that can be spun off of this manuscript, this project. And one of the things I want to do, and this might answer your question, is run the pathway association analysis from Marilyn Warburton to see what pathways show up. So this obviously has some lignin-related traits, but I want to take the same data set and run it through the pathways to see, are there JA-related traits? What other pathways can come up and how strong are they? I'm not sure if that answers your question or not.
Yeah, I think that would be a neat addition to this. But yeah, there's all sorts of analysis to be done.
Yeah, and one thing I didn't say is I don't know if Mike Colometes is on the link, but I've used some of Mike Colometes I screened some of Mike Colometes as mutants for JA, and they are much more susceptible. So JA is obviously very important. I'm very interested to see if the brassinosteroids are important. So there's a lot else that's going on too, and with this particular manuscript, the lignin really come out to play hard. Yeah, so I don't know. It'd be nice to see how important the lignin traits are versus JA or brassinosteroids or other defense mechanisms.
Yeah, for sure.
Okay, and then Ed had another question.
Yeah, Judy, I was just kind of wondering about, do you know anything about what's going on with like brown midrib or other lignin pathway genes in the roots? And, are they under separate expression control, and how do they affect root degradation?
That's a great question. Talking first about the brown midrib mutants, they're a hot mess, and I don't know how anybody would grow them without spraying them for foliar for fungicide. Germination rates. So we don't actually seed coat our seed when we put it in the field. And if you look at the germination rates on the brown midrib mutants, it's significantly decreased compared to wild type. So there's, even in the field, even a germinating seedling has a harder chance. So I imagine that it does something to the roots as well. I have not looked to see what these do. The HCT11 gene has been implicated for UVB protection, UVB resistance or susceptibility through Rory's group, but also gray leaf spot expression, drought, and heat stress. So that might be an indication that there's something in the roots if they're implicated in drought and heat stress. But I haven't looked specifically at them through any other mechanism, through any other data set. But I would think that the brown midrib mutants would have a poor root structure or poor root defense capability.
Yeah. Sounds good, or sounds reasonable. Not good, but yes.
Yeah, there's a reason people grow them, and especially in the last 10 years, but they need fungicide. They do.
Okay, and another question from the Buckler Lab.
Yeah,
That's a good question. Oh, sorry.
No, go ahead. Go ahead.
No, you asked. I just looked at the question. I don't know if that's what you wanted to ask.
Yeah, that was my question of, have you looked into the overlap between HCT11 and the other QTL to see if true biological pleiotropy exists there?
So HCT and the other QTL for, can you still see my screen?
Yeah, that one you're circling.
Yeah, so that's northern leaf bite, gray leaf spot, and lesion mimic. So that makes a lot of sense that there is an overlap here, because these do overlap. And there's expression analysis for gray leaf spot that has in the Gomez Cano paper et al. That I keep raving about because it is really that good of a paper. That shows that gray leaf spot is implicated through expression. The lesion mimic from Peter Ballincurdy's group, this QTL is interesting because he also found other HCT genes involved in lesion mimic, in the lesion mimic phenomenon, HCT1 and 2. And so this is HCT11 that hits right underneath it. So this to me is just a really good pleiotropic interaction. Stock strength, I don't see any stock strength QTL through here, but this GWAS or expression might show something different than the joint linkage mapping. Does that answer your question?
I think so, thanks.
Thank you.
Are there any other questions for Judith? Okay. If that's not the case, then thank you all for joining. Thanks, Judith, for presenting, and see you all next week.
Thank you very much. I really